In the previous video of this series about exocrine glands, we learned how to classify them by duct shape. Here, we will continue to address the classification of these glands by shape, but now referring to the secretory portion. We will start with the answer to the second of the 5 questions made on video 2. And then we will learn how to identify the secretory portions of exocrine glands in histological section slides. The third question will be addressed in the next video, and afterwards we will move on to the remaining questions. We can associate 2 figures to the shape of the secretory portion of exocrine glands: a tube or a sphere, both hollow. The histological terms used to make reference to these figures are a little more refined: tubular and acinar or alveolar. On the wall of the large intestine, the arrangement of the tubular exocrine glands is clear. Each one of these tubes, formed by simple columnar epithelium and supported by loose connective tissue, is an intestinal gland, whose cells are organized as shown on this schematic drawing. Notice that the intestinal glands are straight, different from what we find when we observe, on a skin slide, the eccrine sweat glands, which are coiled. Due to the fact that the histological section provides a 2-dimensional view of a 3-dimensional structure, what we see are several fragments of secretory tubules formed by large columnar or pyramidal epithelial cells. When cells of the secretory portion are organized in a spherical shape, they may receive 2 designations: acinus and alveolus. An acinus has a narrow lumen, which can barely be identified on a slide of a parotid salivary gland, for example, since the secretory epithelial cells occupy almost all the sphere. An alveolus, on the other hand, has a wide lumen, being easily identifiable on a prostate slide. However, you can find the terms acinus and alveolus used as equivalents in some books. We can thus easily see the alveoli with wide lumens on the prostate slide. Still, we also find secretory portions in tubular shape. This happens because the prostate is formed by tubuloalveolar glands, which means that the secretory cells are organized in both tubules and alveoli. Similarly, there are glands which have tubules and acini at the same time. That's the case of the sublingual and submandibular salivary glands, examples of tubuloacinar glands. We have seen in the previous video that the salivary glands are compound, which means that their main duct is divided in many other ducts. One can notice that both in the schematic drawing and the histological slide, the secretory portion is well developed. The schematic drawing shows that the secretory cells are structured in tubules which connect to spheres. On the histological section, we identify these tubules and these acini. We have seen then that in relation to the shape of the secretory portion, the multicellular exocrine glands may be tubular, acinar, or alveolar, or even tubuloacinar or tubuloalveolar. In the next video, you will see that the tubules, acini, and alveoli may or may not be branched, thus completing the classification of exocrine glands by shape. We shall then move on to the classification of this type of gland by secretion.